Hi micro folks, we're just doing results of our chapter 11, aseptic transfer of bacteria. So folks, you'll recall that we transferred bacteria into sterile broth. So here's some of our sterile broth, and notice it's transparent, and that indicates that no cells are present. Now, this was when I was going to inoculate with E. coli, and I think I forgot to do it, so it's good. It shows uninoculated sterile medium. Now, however, folks, if I had inoculated this with E. coli and I incubated it and I pull it out of the incubator and it's transparent, I'm going to check the bottom to see if there's a pellet. I'm going to shake it to verify that it's not cloudy or turbid. Okay, so recall that cloudiness or turbidity is a sign that cells are present, and usually we use it as an indication that our culture grew. So had I inoculated this E. coli and it came after incubation, it's transparent, I would call that a failure of transfer, and this does happen. So we'd want you to know what are reasons for failure of transfer. Well, one thing is maybe you forgot to inoculate it, right? The second thing is maybe your stock culture is dead, right? The cells that you transferred are dead. Another possibility is maybe if you transferred from a plate, maybe your loop was too hot and you killed the bacteria. Another possibility is you transferred them into medium in which they can't grow. And I think that might have happened with one of our other experiments. Or you incubated them at a temperature at which they can't grow. So those are all reasons for failure of transfer, and that's often a lab exam question. Okay. Do you remember, you guys, just again as a safety reminder, these tubes, we placed a paper towel in a can, and we sprayed the paper towel with disinfectant. We incubate our tubes in the cans because we don't have enough room in the incubator for test tubes. And remember, you guys, safety thing, we always barely loosen the cap so they can vent. Okay, so that's our, this was our Citrobacter, nice and turbid, so we knew our Citrobacter grew. A disadvantage, you guys, of broths is just looking at this Citrobacter culture. Can I be sure it's pure? And the answer is no. Just looking at it, all I know, cells are present, so I can't know if it's pure or a mixed culture. And this was one of the disadvantages that Pasteur's lab had when they were competing against Robert Koch's lab trying to prove the germ theory of disease. In proving the germ theory of disease, you have to have pure cultures. And since Pasteur's lab, all they had were these broth cultures, they couldn't guarantee their cultures were pure when they did their experiments. And you'll recall Robert Koch's lab, his lab was one that introduced the use of agar to make solid media. And because he was able to have solid media, to have agar plates, he could guarantee he had pure cultures, and thus he won the race to prove the germ theory of disease. One more thing, folks, I forgot. Let me just show you. This is out of the photo atlas, page 27, and I'll try to get also some pictures on a photo atlas. Here's patterns. Oops, can't do this very well. Here's patterns of growth that we would expect to see. So our Citrobacter here, before I shook it, it was already turbid, and that suggests that possibly you have motile. Right? They're motile organisms, right? So they're swimming throughout the medium. These next two here, folks, it looks like they're transparent, but if we picked up the tube and looked at the bottom, we'd see what's called a pellet or a button. And this forms because even though the bacteria are little, they still have mass. And if there aren't motile, they're going to gradually settle to the bottom of the tube, forming that button or that pellet. And that's why we always want to check the bottom of the tube. If we think we had a failure of transfer, we check the bottom of the tube for a pellet or a button, and then we gently tap the tube to resuspend the cells. And often we'll be pleasantly surprised to see that the cells did grow. They just settled to the bottom. And usually that's because they're not motile. And then over here, you guys, this is really cool. You'll see this layer. It's actually a type of biofilm, like in our kombucha tea. You'll see this layer of cells at the air broth interface. That's called a pellicle. And often that's a huge advantage if a bacterium can carry out aerobic respiration, or maybe that's all they can do. It's a huge advantage if they can form a pellicle at that air broth interface, because that's where the maximum amount of oxygen will be. You'll recall, folks, that oxygen doesn't diffuse well through aqueous water-rich solutions. So as we go deeper and deeper in the tube, there's going to be less and less oxygen. So if you need oxygen to carry out aerobic respiration, forming a pellicle So you can have maximum access oxygen will be a huge survival advantage. Okay, folks. Then another transfer we did is, you recall, we did a stab and streak. We used our inoculating needles, and this little guy is Citrobacter again. So we used our inoculating needles. We stabbed the butt to see if our little guys could grow anaerobically. And I know it's hard for you to see, folks, but I can see there's definitely colonies. There's definitely cells growing in the butt. And then we withdrew the needle and we streaked the surface. And again, folks, I apologize. I know you probably can't see this, but I've got colonies growing on the surface. So I know my microbe can grow aerobically and can also grow anaerobically. And another cool thing, folks, and again, doggone it, I wish I was better at this. I wish I was a good cameraman. In the butt where the Citrobacter is growing, there's cracks and bubbles. And that's because as it's growing anaerobically, it's carrying out fermentation and making lots and lots of gas. And those gases, you guys, they could be molecular hydrogen or carbon dioxide or both. So we know our Citrobacter is a good gas producer. So we'll see later, folks, this is one reason, like when we incubate our tubes in those cans, remember we're going to loosen the caps, but we always want to put a paper towel in the can and spray it with disinfectant, because we'll see later sometimes there's so much gas produced in these big butt slants that the slant will be blown apart and it will blow the agar up. And sometimes there's so much gas pressure, the agar can squeeze between the cap and the glass lips, and you end up with bacteria and agar contaminating the outside. So usually if we see that that's happening, we'd want to spray the tubes first with disinfectant. And again, we put that disinfectant soap paper towel in the can just to try to help kill as many bacteria that escape as possible. And then, folks, this last one is, it was an E. coli, and we just did a regular transfer to a regular agar slant. And you recall this is, we transferred the bacteria to the surface of the slant. And here we can see they're growing very happy. I don't think you can see because I'm not doing a good job here, but I can see that we've got really good growth. The growth where the cells are growing is in that nice S-shaped pattern. Remember, we kind of slithered our loop up the slant. Okay, so we had good growth with the E. coli as well. In contrast, you guys, these are the uninoculated, right? So here's my uninoculated big butt slant. I can see there's no growth in it. And here's my regular slant, and again, I can see there's no growth. All right. Now, folks, one thing I forgot to do when we were looking at the airborne plates, usually what we do is we introduce you to ways to describe different types of colonies, right? And this is best done when you're using agar plates. On a slant, very often the colonies aren't isolated, so it's hard to distinguish them. So these are some of the features that we use to describe our colonies. So we can describe elevation. We can describe we can describe the margin, and the margin, you guys, another name for the margin is the edge or the border. So you see different examples of margins or border here. We can describe the shape of the whole colony here, so we can see different types of shapes there. We can describe the texture of the surface of the colony. And folks, I don't know if you're seeing this, but I keep getting these join Xfinity things popping up. Sorry. We can describe the optical properties of the colony by holding it up to the light. Does light shine through? In which case we say it's transparent like glass. If a little bit of light shines through, we call it translucent, and if no light shines through, we call it opaque. So again, folks, we use these colony characteristics to describe specific colonies. And again, different microbes should form different types of colonies, and we would presume that the same microbes should form the same types of colonies, right? And in the photo atlases, you guys, and in the PowerPoint, I'll try to give you some examples of some of the cool different types of colony growth. And I think I had, yeah, this was a cool one, you guys. So this is an example. I think these are airborne plates. Oh no, this is soil. Oh, we should do that, you guys. We'll do that. So these are nutrient agar plates, which is another example of an all-purpose media. And what they did is they microbes from soil. Maybe they mix the soil with a little bit of water, and then they spread the water containing the microbes out over the surface of the plate. And you can just see this incredible variety, different types of colonies, different colors, different sizes, different shapes, different borders, different optical properties, different textures, right? So I should have done that. I should have done a soil sample. Maybe I can still do that at home, and I can show you guys on another movie. Okay, so I think that does it, you guys, for chapter 11. So we'll stop it here.